My name is Björn Schumacher. I'm directing the Institute for Genome Stability in Aging and Disease. And we are very interested in finding out how aging works. So we are specifically interested in how the building blocks of our basic information influences aging. The DNA encodes everything in the cell, in an organism. It's all the information. And the DNA can be damaged, and it undergoes damage all the time. And so it needs constant repair because it's the information content of the cell, so it cannot be replaced. And that sets DNA aside from any other biomolecule that we have. It has to be constantly repaired. And when humans are not able to repair the DNA, they can age very, very rapidly. And there are children that already age before their teenage because they cannot repair DNA. And that's what we want to find out.
And is this binary, it either can repair or can't repair, or is there a gradual degradation of repair?
There's indeed a gradual degradation of repair. And what's really interesting is that if you look for longevity or even immortality, nature has already invented that, and that's our germ cells. Our germ cells carry forward the genetic informations over generations and generations. Our germ cells that we have are already over 200,000 years old in modern Homo sapiens, and they repair very, very, very well. But our body, which we also call the soma, our body cells are much more restricted when it comes to repair because we are only need to exist for a few decades and be repaired for a few decades. So our repair in our body is really much, much restricted compared to our germline repair. And what we are trying now to do is to learn how germ cells repair so well and then transfer this ability to the body cells to make our genome more stable and delay the aging process of our body.
And could you tell me about some of the more exciting results that you found in this work?
So what we found is the first master regulator of DNA repair. It's called the DREAM complex. And what it does, it represses a lot of the DNA repair factors in our body cells. And now, if we can inhibit this DREAM complex, then we can elevate repair. Now, a lot of more DNA repair factors are made in our body cells, and they can then repair better and reduce the cancer risk because DNA damage can lead to mutations that cause cancer. We think we can reduce the cancer risk, and we can extend the durability and integrity of our genomes. So that there we have for the first time a really causal mechanism that we can intervene in order to target the aging process and have better, healthier lives.
And is this DREAM complex shared among species, or is it human specific?
The DREAM complex is very ancestral. We are studying in the lab a nematode worm, a very simple worm, 1 millimeter long, Caenorhabditis elegans it's called. It's the nematode worm. And there we discovered this link between DREAM and DNA repair, and this DREAM is exactly the same factor that we have in humans. And so that's why we can translate our studies in the simple worm to humans, and now also study in mice and how far we can have in a mammal really a health benefit of improving DNA repair.
And what kind of results have you seen by suppressing the DREAM complex in simpler life forms?
In simpler life forms, they make the animal resistant to DNA damage. They can repair all sort of different DNA damage much better than a normal worm. And then we validated that in a mouse that has a degenerative pathology. In this case, it was a degeneration of the retina, a very important age-related disease in humans. And we could prevent that by inhibiting DREAM pharmacologically and then preserve the function of the eye. And so that's why we can take our very basic discoveries in the worm into a higher animal, into a mammal, on the path to really develop that for the benefit of humans.
And are there small molecules that can inhibit the dream complex that you found?
Indeed, and that is really what is so exciting about it, because the dream complex is assembled when a specific kinase is operating on the complex, and this kinase we can inhibit pharmacologically. So we have these inhibitors that can inhibit the kinase, and the inhibition of the kinase leads to disassembly of the dream complex, and then the de-repression of the DNA repair genes. So repair genes are then induced, and so we can really pharmacologically, with small molecules, achieve better DNA repair.
So where do you see your field progressing over the next 5 to 10 years?
I think the real breakthrough is that for the very first time, we can now target the real mechanisms of aging. Because thus far, most of the interventions are metabolic interventions that can delay aging to some extent, but the interventions have not thus far targeted really the most fundamental root causal mechanisms of aging. And their DNA damage is really at the hierarchy the most upstream event, because it has consequences on everything in the cell. And that's why we can zoom in now on the causal mechanisms of aging and really target aging where it first is driven by. And that, I think, we will see real breakthroughs in combination to also, as we here learn at the Biomarkers of Aging Conference, that we can make biological age measurable.
And given that we're at the Biomarkers of Aging Conference, what do you think is the most important biomarker to pay attention to?
I think we need the plethora of biomarkers, and that's why omics technologies are so important for the biomarkers. Traditionally, there were like a few markers, a few diagnostic markers, and I think now with the field of omics technologies, so where you can look at many different changes in a cell, many different changes on molecules, you can now really get a plethora of data, and the combination of so many different data points give you then a very good assessment of age, and increasingly of biological age, because biological age can differ from chronological age. Some people age faster, some slower, some get sick earlier, some have a specific disease susceptibility, and we can make this all measurable. I personally think that transcriptomic clocks, which are based on gene activity, are very exciting, because they tell us a lot more than just the measurement of age. They tell us a lot more about what is going on in the cell, what processes are changed. And so I'm very excited about the developments on the so-called transcriptomic clocks. And I also find it very exciting that we learn now why clocks work so well, because aging is not a program. Aging is the default. And aging is increase of chaos, increase of dysfunction. And so we showed that recently that stochasticity alone can give us a very accurate prediction of age. How much randomness, how much noise have expanded on our molecular parameters.
That's fascinating, and your work is just very exciting. If people want to learn more about what you do, where should they look?
So we have, of course, a website, and everybody is very much welcome to get more information from our website at the Institute of Genome Stability in Aging and Disease, and also at the Cologne Excellence Cluster for Aging Research, which is one of the largest agglomerate of aging research in Europe. And of course, going to YouTube, watch some of the podcasts and the videos where we also explain to the general audience what we learned about aging. I very much invite your audience to join us and check out what we are doing.
Thank you so much for your time.
All right, it was a pleasure.
Pleasure.
Thank you.